Now,
Yes, now we see.
yes, you can see it. Okay, we start the immune system. You know that this is the body's response to disease and injury, and it consists of molecules, cells, tissues that mediate the resistance to infections. And we have the two responses, non-specific or innate immunity, and the specific response, which is called acquired immunity. The non-specific immune defenses activate on exposure to any foreign substance but react similarly regardless of the type of antigen. They are unable to identify the antigen. The principal component of this system is phagocytic system, phagocytosis. This is phagocytosis. That means the process of ingesting and digesting foreign substances. Phagocyte cells are neutrophils and monocytes. We will repeat this once again. The specific defenses have the ability to recognize the antigen and respond selectively. The components of acquired adaptive immunity are humoral immunity and cell mediated immunity. The cells responsible for these two forms of immunity are lymphocytes, specifically B lymphocytes, T lymphocytes, and so on. We'll talk about it in details. Here you have the components of immune system, all the lymphatic system with lymphatic structures, lymphatic vessels, bone marrow where all the cells are produced, the thymus gland, lymph nodes, adenoid and tonsils, which are lymph structures, and so on and so on, as well as some tissue cells, interstitial fluid as well, and all the other masses of in the intestine and some other organs. So the non-specific external barriers are skin and mucous membranes. If they are penetrated, the body responds with innate immune response. Phagocytic and natural killer cells are included, inflammation, fever as well. And if the innate immune response is insufficient, the body responds with adaptive immune responses. That means cell mediated immunity and humoral immunity. The first line of defense, non-specific defense, designed to prevent infections by viruses and bacteria and include intact skin, mucous and cilia, and phagocytes, plus saliva and tears. This is very important for children because they contain antibacterial enzymes, mm, stomach acid, because the low pH kills the harmful microbes, and all the mucous linings that trap dirt and microbes, and of course the so-called good gut in bacteria, in the intestines. The role of skin. Dead skin cells are constantly sloughed off and makes it hard for invading bacteria to colonize. No colonies can be formed. All the antiperspirants and deodorants contain antimicrobial chemicals, including some antibiotics. For instance, maybe about 60 years ago, when hexachlorophene was used for the first time in pediatric ward, in neonatal wards, it had very good effect on security of skin of the neonates and small babies because of its antibacterial properties. The role of mucus and cilia. Mucus contains lysozymes that are the enzymes that destroy bacterial cell walls, and the normal flow of mucus washes bacteria and viruses off the mucous membranes. Cilia in the respiratory tract especially move mucus out of the lungs to keep bacteria and viruses out. You will see later when we study respiratory system how what a good job are doing cilias in this tract. The role of inflammation. Inflammation is signaled by mast cells which release histamine. Histamine causes fluids to collect around an injury to dilute toxins. This causes a swelling, in inflammation, and the temperature of the tissues may rise, which can kill the temperature sensitive microbes. Inflammation process brings immune cells into the damaged area of the body so they can destroy foreign cells, and they express cytokines. They release, the immune cells release cytokines and regulate immune responses, the so-called pro-inflammatory cytokines from macrophages. They coordinate the inflammatory responses in the body, the big family of tumor necrosis factor. It has 19 protein members and regulates immune response in inflammation, but as well the proliferation, differentiation, and the apoptosis of cells. The role of fever. Fever is caused by toxins on the surface of viruses or is released of the release of histamines by the damaged cells, by the accumulated toxins in the body, and the body pyrogens that signal the hypothalamus. Fever is a defense mechanism that can destroy many types of microbes, helps the fight against viral infections by increasing interferon production. While high fevers can be dangerous in small babies, the central nervous system is not well functioning in the early ages. The function, it is quite well developed, but the function is not quite ripe. So high fevers can be dangerous, but low fevers should run their course without any treatment. Pathogens. Big percentages of disease in humans are caused by infectious agents, different microorganisms that can be bacteria, virus, fungi, protozoa, parasite, even the prions, misfolded proteins, small particles of misfolded proteins of the host. Antigens, most of them are proteins or large polysaccharides from a foreign organism. For instance, from microbes, they can be the capsules of the microbe, cell walls, toxin, caspids, viral caspid, and the flagella, and many others. Non-microbes that serve as antigens may be pollen, egg white, red blood cell surface molecules, serum proteins, and surface molecules from transplanted tissues. Lipids and nucleic acids are only antigenic when combined with proteins or polysaccharides. Hapten means small foreign molecule that is not antigenic, must be coupled to a carrier molecule to become antigenic. Once antibodies are formed, they will recognize haptens. The antigens have this special part, small part of the antigen that interacts with an antibody and is called epitope. And any given antigen may have several epitopes. You see different forms. And each epitope is recognized by a different antibody. You see A or B or C. We come to the second lines of defense. They involve the white blood cells, white, the white line in the bone marrow, white blood cells. The non-specific response is when invading pathogens are targeted by the macrophages, and the specific response comes when lymphocytes produce chemicals called antibodies that target the specific pathogens. Macrophages. Macrophages are found in organs, not the blood. They are produced in bone marrow as monocytes, and they are called macrophages once they reach organs. Usually, the name is based on tissue where they reside, and you have heard about alveolar in the lungs, Kupfer in liver, microglial in brain, or osteoclasts in bone. They live long and initiate immune responses as they display antigens from the pathogens to the lymphocytes. They're the connecting, they're cells that present antigens. The antigen presentation signals other cells of invasion, activates T cells to duplicate, and produce interleukin that promotes other cell activity. It is a very already very well known process, very interesting and very complex. Here are phagocytes and their relatives, monocytes, macrophage, dendritic cell, and all the white cells plus mast cell. What is the difference between, I think I had a slide about dendritic cells, but I can't see it. Yes, it comes later. Uh, Maybe I will talk on it now. Dendritic cells, as relatives to macrophages, they were discovered recently, you see, 15 years ago, and they are professional antigen presenting cells. Antigen presenting cell. There are several types. You must not remember all the details, but usually they are circulating dendritic cells, and they can be myeloid, lymphoid, plasmacytoid, follicular, dendritic cells in lymph follicles, interstitial. They populate organs such as heart, lung, liver, intestines, the Langerhans cells. They are found in skin, and they are interdigitating dendritic cells in the T cell areas of lymph nodes and thymic medulla. What is the difference between macrophages and dendritic cells? The macrophages, you know already, these are white blood cells that surround and kill microorganisms, remove dead cells, and stimulate the activity of all the other cells in the immune system. Dendritic are special type of immune cells that boost the immune response, showing antigens on its surface to other cells of the immune. So this is the main difference. They show the antigens to the other cells. The macrophages occur in the stationary form in tissues or as a mobile white blood cell, especially at the sites of infection, while the dendritic cells are not in the blood, they occur in tissues and the skin. Macrophages initiate the inflammatory process in tissues, while the dendritic cells activate upon inflammatory signals. And the macrophages do not die after achieving their effector function, while the dendritic cells die after they achieve the effector function. So the role of phagocytes is to seek and destroy invaders. Some also destroy damaged body cells, and they are attracted by the inflammatory response of the damaged cells. Here is the detailed process of first you have chemotaxis and adherence of microbe to the phagocyte. It is ingested by the phagocyte and a phagosome is formed, a phagocytic vesicle. Then there is a fusion of the phagosome, this phagosome vesicle, with lysosome to form a phagolysosomes, and the ingested microbe is digested. There remain partially um, residual bodies that go out of the phagocyte or on the surface of the phagophyte, and they will be presented later. This is the electron microscopy of this process, you see. And remember that pus is always an accumulation of dead phagocytes, destroyed bacteria, and dead cells. You can have this process all over in the organism after a real struggle between invaders and our defense immune system. Here is the development of dendritic cells. You remember that hematopoietic stem cell gives two daughter cells, the common myeloid progenitor and common lymphoid progenitor. From the common myeloid progenitor come everything, all the other cells that are produced come from this cell, even erythrocytes and thrombocytes. You have here the different dendritic cells, and the monocyte who going to the tissues will be called myeloid dendritic cells or macrophage. Only the lymphoid cell line is differentiated alone. From the common lymphoid progenitor, we have the lymphocytes, the production of lymphocytes. Here we have the animation of antigen presentation. After the phagocyte is eaten by the phagocyte eats the bacteria. These parts of the undigested goes to the surface and it is presented to the helper T cells. They are very important, you will see later, and they are activated in this way. It is done animation for small children, but it's better to remember this image so you will understand very well the antigen presentation. The organs of immune system are divided into primary lymphoid organs and secondary lymphoid organs. The first one include bone marrow and thymus gland, which is here, and the maturation site in bone marrow. The secondary lymphoid organs are the spleen, the lymph node, the mucosal associated lymph tissue, and gut associated lymph tissue, very important, especially in small children. Here you have the thymus, the beloved organ on top of heart. It is in the anterior mediastinum, and it is very important in children because you see in infant it weighs 70 grams and only 3 grams in adults. The best differentiation is in the early pediatric age, and it reaches the maximum size during puberty, and after that comes an involution. So we don't have it an older age. Here is the lymphatic system, vessels, and follicle nodes. You will see the schemes with more details. Here is the lymph node with characteristic germinal centers where B lymphocytes proliferate or stay as a depot. What other? The lymphatic artery and a lymphatic vein and the post-capillary venule to do the connection between lymph, lymph and blood. Here you have the mucosal associated lymphoid tissue. The mucous membranes in our organism is about 400 square millimeters, and this is the most common pathogen entry site. So it has to be protected, and this is done by mucosal associated lymphoid tissue. They have these mucosal cells have a unique architecture that allows the antigen entry, and the most organized of these all these membranes are the Peyer's patches and lamina propria in the intestines. In the intestines. Here you have the a Peyer's patch. Very important are these M cells. This is microfold cell. The name is microfold cell. They are located in the epithelium that covers the mouth, the mucous membranes, and actively transport from pathogens from the lumen. Here you have the intestinal lumen. Actively transport the lumen and antigen to the underlying lymphocyte follicles. So the immune response is initiated. Here you have the same scheme of mucosal epithelium that consists of these M cells transporting the antigens in the organized lymph follicles that produce, for instance, immunoglobulin A in that place. The key players of the immune systems, like cells, are the white blood cells, leukocytes, and especially the polymorphonuclear granulocytes. When we talk about white blood cells, we leukocytes, we mean granulocytes, the group of granulocytes, and the lymphocytes, monocytes. Granulocytes have more than one nucleus, more than one, while lymphocytes and monocytes have only one nucleus. Maybe you remember this. Here you have two kinds of monocytes with sometimes they have very strange nucleus. These are, and the difference in the granulocytes is done upon the color they receive after treating with Gimsa stain. The neutrophils this have this neutral color of the cytoplasm, while the eosinophils are bright red color and they have too many granules like red caviar. They look like red caviar, and usually the nucleus are only two, and usually they're connected with a small bridge. So sometimes they look like spectacles, the eosinophils. Basophils are, you know, that they are a very small number in the blood formula, and especially in children, they are no more than 1% in 100 cells. And so these are the key players of granulocyte have phagocytic function, and lymphocytes who may be T, helper T, B lymphocytes, and the natural killer cells, monocytes or macrophages, and dendritic cells. Plus another player, the antibodies that will come after the activation and special proliferation differentiation of B cells. They will turn into plasmocytes and memory cells. Sorry. All these cells, you know, they are in lymph nodes, vessels, bone marrow, thymus gland, spleen. Their life cycle passes through the spleen, and usually they are destroyed in the spleen, and some of them in gastrointestinal tract to attack body foodborne pathogens, or in the respiratory tract for airborne pathogens. B cells, B lymphocytes, they recognize antigens, proliferate, and produce specific antibodies. The antibodies travel to the blood, to the lymph, the lining of gut and lungs, mucosa lining. They differentiate into plasma cells to produce more antibodies, and the number of the plasma cells goes down after a few weeks. They can differentiate into memory cells to keep antibodies in supply for activation from second encounter by same antigen. Memory cells divide rapidly. As soon as the antigen is reintroduced, it may be destroyed so rapidly they divide that the antigen can be destroyed before any symptoms show when they encounter for the second time. And there are many more memory cells than clone cells. Here's again an animation on activation of B cells, but this is important to remember. Here you have uh, differentiation of a B cell, the resting lymphocyte recognize a specific antigen, for instance, antigen A, they recognize it, and they start to clone the resting lymphocytes. Here you have on which day the changes are seen in the cell. Here, ribosomes start to produce, and the antibody molecules are synthesized in the endoplasmatic reticulum on day 3. They proliferate more and more and start on day 3 to form the antibody molecules. On day 5 is produced the first plasma cells and antibodies are secreted. This lymphocyte does not divide, does not differentiate because it can recognize only antigen B. Each lymphocyte recognizes specific antigens. And at the last stage is differentiating into plasmatic cells and memory cells as well. Here is the last differentiation. A very beautiful picture. I like very much this development and activation of B cells. It is like a cosmic image. So I decided to show you T lymphocytes. Mature T cells have T cells receptors, which have a very similar structure to antibodies and are specific to one certain antigen. They are activated when the receptor comes into contact with the antigen on a macrophage membrane or an invaded body cell, destroyed destroyed or ill body cells. The T cell receptor recognizes only antigens combined with major histocompatibility proteins on the surface of cell. You know, maybe you remember that the histocompatibility cells class 1 are found on all cells and class 2 are found only on phagocytes. So, after activation, T lymphocyte divides to form T helper cells. They secrete cytokines, very important for the activation of B cells, help the B cells to divide, stimulate macrophages. Most of them have this glycoprotein on the surface of the cells that is a marker, molecule marker, CD4 plus. Another subdivision are the cytotoxic T cells or killer T cells. They kill body cells that display antigen and remain in body. And the regulatory, now they are called regulatory. They were formerly known as suppressor T cells, but they are now T regulators. They modulate the immune system, maintain tolerance to self-antigens, this is important, and prevent autoimmune disease. The regulatory cells are immunosuppressive and generally suppress or downregulate induction and proliferation of effector T cells. They express other biomarkers, plus CD4, they have some other biomarkers. The T helper cells, once again, we will repeat this. They express the CD protein on their surface and are presented with antigens class 2 molecules. and start to secrete cytokines that will regulate and assist the immune. They have the central role in the immune process. Here is a more detailed scheme of presenting antigen and of the helper T cell with its receptor, and then it activates the B cell. Now, if all the cells are not properly educated, they will be negatively selected. The education of T lymphocytes is very important, and if they are preparated in bone marrow surface preparatory school, they must absolutely pass the Timus University, and after that, they will easily recognize the antigens that they will eventually meet, for instance, 3 meters along the road to the left. The thymus induces the death of those T cells that cannot recognize antigen MCH, MHC complexes, and those that react with self-antigen, major histocompatibility complex, and of course pose a danger of causing autoimmune disease. And you have to know that more than a very big percent, more than 95% of all thymocytes die by apoptosis in the thymus without ever reaching maturity. So the students that leave the Thymus University are only 5%, but they are very well educated. The cytotoxic T cells are destroyed target cells. Most of them do not have the biomarker CD4. They recognize antigens on the surface of all cells. They kill host cells that are infected with viruses or bacteria. They recognize and kill cancer cells. They recognize and destroy transplanted tissue because they release protein called perforin, which forms a pore in the target cell and causes lysis of infected cells. And when the stimulating antigen is gone, they undergo apoptosis. Here is the T cell binds to an infected cells, and the pores that are formed in the infected cells through this perforin, which makes the pore. So ions and water enter the cell, and it is lysed the infected cells. Now we come to antibodies. They are also known as immunoglobulins. In fact, they are globular glycoproteins, polypeptides, which form heavy and light chains held together by disulfide. Here you have the disulfide binding bridges, and heavy light and two heavy chains. Each antibody has two absolutely identical binding sites. They are called very variable regions, and the order of amino acids in the variable region determine the shape of the binding site. How do they work? Some of the antibodies act as labels to identify antigens for phagocytes. Some work as antitoxins. They block toxins, for instance, from diphtheria and tetanus. Some of them attach to bacterial flagella, making them less active and easier for phagocytes to engulf. Some cause agglutination, clumping together of bacteria, making them less likely to spread. There are now five different classes of immunoglobulins. You know them: EGA, EGE, EGD, EGM, EGG. Some words about each one. The EGG is a monomer, has a monomer structure. It is with a very high percent, the highest of percentage in the serum. You see 80%. It is found in blood, in limbs, in the intestine, along the longest half-life, 23 days. Fixates complement transferred through to placenta, and the known functions till now are that enhances phagocytosis, neutralizes toxins and viruses, protects the fetus and newborn because of this ability to transfer the placenta. EGM pentamer structure. You see the percentage in the serum, much more, much less. How is the located in blood, lymph, B cell surface, half-life only 5 days, fixate complement, but there is no placenta transfer, and it is known as the first antibody that is produced during an infection. Effective is against microbes and agglutinating antigens. The immunoglobulin A class, a dimer structure, 10-15% in serum. It is found in different secretions like tears, saliva, in the intestine, in milk of the mother, blood and lymph as well. Half-life only 6 days, does not fix complement, and there is no placenta transfer. One of very important function is the localized protection of mucosal surfaces, provides immunity to the infant digestive tract. EGD is a monomer structure. It is with a very small percent, and there's the smallest percent, I believe, in serum antibodies. It is in on B cell surface in blood and lymph, 3 days half-life, no fixation, no placental transfer. The function in serum is still unknown, but on B cell surface it initiates immune response. EGE, a monomer structure. This is the smallest, in fact, percentage of IgE. It is bound to mast cells and basophils throughout our body, in blood as well. 2 days half-life, no fixation, no transfer. It acts in allergic reaction and possibly lyses worms. They have the eosinophils, that contain too much immunoglobulin E in the granules. They can kill worms. Now, immunoglobulins in children. The newborn receives enough amounts of maternal IgG, which confers the immunity up for about 3 months. This is immunity against antigens to which the mother has been exposed. All the antigens she has met and she has contacted, she has immunoglobulins G in her and they are transferred to the baby. During this time, at month 3, the infant begins to synthesize his own IgG, and at the age of 9 months, about 40% of the normal level in adults is reached, while the uh, no, I'm sorry. This is not about EGG. The 40% are reached at 1 year of age, 1 year of age, and it continues to grow later. Significant amount of IgM are produced at birth adult levels, reached at 9 months. They are produced, you see, almost starting um, from conception, it is marked. But in fact, the adult levels are reached at 9 months of age, almost. And the production of the other immunoglobulins like IgE and IgG and IgE is much more gradual. Maximum levels are not attained until early childhood. Cytokines, some words on cytokines. These are groups of proteins made by the immune system and they act as chemical messenger. Very different types: chemokines, interferons, interleukins, lymphokines, the tumor necrosis factors, we talked about this. They can act alone, they can work together or work against each other, but ultimately the role of the cytokines is to help regulating the immune system. The complement system. It is part of the innate immune system, can be recruited and brought into action by antibodies generated by the adaptive immune system, enhances or complements the ability of antibodies and phagocytic cells, promotes inflammation, and attacks the pathogen's cell membrane. It consists of 30 about 30 proteins and protein fragments found in the blood, or they are synthesized by the liver. They circulate as inactive precursors and they account for about 10% of the globulin fraction of blood serum. When stimulated by one of several triggers, proteases in the system cleave specific proteins to release cytokines and initiate an amplifying cascade of further cleavages. The activation can be initiated by 3 different biochemical pathways, just to have some impression: the classic pathway or alternative or the so-called lectin, but these are specific details. Maybe if you work immunology or laboratory, you will remember them better just to have an impression. The terminating of the immune response. The turning of the immune system is as important as turning it on. Turning off is important as turning it on. When the invader is destroyed, the helper T cell calls a halt to the immune response. Memory T cells are formed, which you understand they can quickly divide and produce cytotoxic T cells to quickly fight off the invader if it is encountered again in the future. The inhibition and resolution of the immune responses are important to prevent autoimmunity and reduce ongoing inflammation. The duality of the immune system, that means humoral or antibody mediated immunity, is involved in production of antibodies against foreign antigens. They defense against bacteria, bacterial toxins, viruses that circulate freely in body fluids before they enter the cells. They also cause certain reactions against transplanted cells. While humoral, the cell-mediated immunity or cellular immunity involves specialized set of lymphocytes that recognize foreign antigens on the surface of cells and tissues. Bacteria and viruses that are inside host cells and are inaccessible to antibodies. Fungi, protozoa, and helminths, cancer cells, and transplanted tissue. Here you have more detailed scheme of the duality of immune system. The apoptosis or the programmed cell death, this is the meaning of apoptosis, is needed because human body makes 100 million lymphocytes every day. If an equivalent number doesn't die, leukemia will develop. So B cells that do not encounter stimulating antigen will self-destruct and send signals to phagocytes to dispose of their remains. Many virus-infected cells will undergo apoptosis to help prevent spread of the infection. Of course, the optimal effectiveness we have in balanced immune systems. Otherwise, we may have immune overreaction or immune underreaction. And the overreaction as internal treat is the autoimmune problem, while the external treat of overreaction are the allergic reactions. The underreactions are infection and disease. The overreactions cause cell damage through excessive immune response to antigens. This is the hypersensitivity, overreaction to infectious agents, or allergy, overreaction to environmental substances, and autoimmunity, overreaction to self-proteins. The terminology that we use very much in talking about immunity is the antibody titer. That means the amount of antibodies in the serum, or the pattern of antibody levels during infection. The primary response, you know already, after initial exposure to antigen, no antibodies are found in serum for several days. You saw that they start to be produced on day 5. And then follows a gradual increase in titer, first immunoglobulin M, and then immunoglobulin G is observed. Most of the B cells become plasma cells, but some of the B cells will become long-living memory cells. Then follows a gradual decline of antibodies. Now, this is a problem that is very much discussed in connection with the COVID infection. This is once again we will repeat the normal development of the immune system. After birth, the infants are born sterile, but then they are rapidly colonized by organisms and then started to be challenged by waves of transient pathogens. So the cellular immunity is active from birth. The neutrophil count is relatively low, but infants are able to respond to bacterial infection with leukocytosis. The number of leukocytes is increased. Humoral immunity is less well developed, but the maturing system is initially supported by transplacental maternal derived IgG antibodies and by breast milk factors that include IgA, and as they can produce IgM as well in response to infection. In normal immune system, the waning of maternally derived IgG is matched by gradual enhancement of endogenous production with a nadir, a peak of circulating IgG levels at age 2-3 months. Endogenous antibodies production is accelerated during the preschool years. Children before the age of 5 average 6 to 12 infections each year. 12 infections, that means that each month they can be ill, and this is specific for the pediatric age. The loss or inadequate function of the different components of the immune system will cause immune deficiency. It can occur in any part or state of the immune system. So we can start with the physical barriers, phagocytes, B lymphocytes, T lymphocytes, complement, natural killer cells, and the immunocompromised host has an impaired function of immune system and is at high risk of infection. The primary immunodeficiency states are intrinsic are less than 10%. They are not big percent. And they may be missing enzymes or missing cell type or non-functioning component. They're usually congenital and they manifest since early age, while the biggest percentage is of secondary, and you will see next time the lists of all these immune deficiency state. I think we can stop here. So next time to talk about different immunodeficiency state barriers and some mucociliary defects and so on. I would like to hear from you. Are we going to have the next Tuesday lecture? Because uh, you hear me, okay? Are you hearing me?
Yes,
Yes,
yes.
doctor.
Yes. Next Tuesday is the 7th of December, and on the next day is your holiday, the students' holiday in Bulgaria. So, are you going to celebrate to be absent or something of the kind, or shall we have our lecture at the same time?
Uh, we can have election. It will be online anyway.
Okay, okay. So, next Tuesday, we continue with immunodeficiency conditions, and I wait for your invitation at 5:20 or something of the kind. Hmm?
Hmm.
Okay,
Yes.
Sutili?
Yes,
Okay.
Professor.
Hmm. Nice evening to everybody.
Have a nice
See
evening.
you next Tuesday.
Bye bye.